All right. Well, Steve, if you're ready, we'll go ahead and get started. I see that people are continuing to join, but we want to make a full use of the hour that we have with you. So I'll start by welcoming everyone. Good afternoon. My name is Shanita Hughes Halbert. I'm at the Medical University of South Carolina. And I'm pleased to welcome you to the Disparities Research and Methods Seminar Series that's co-sponsored by Region 1 South in the Geographical Management of Cancer Health Disparities, which is a region that includes North Carolina, South Carolina, and Tennessee for the purpose of integrating and integrating collaborative efforts and developing new collaborations around minority health and cancer health disparities. A particular focus of our activities within Region 1 South and across the entire GMaP program is to enhance the professional development of all of us who are doing cancer health disparities research. And this seminar series is one of the ways in which we do that. Our program is also sponsored by the Hollings Cancer Center, as well as the NCI Community Oncology Research Program. So we're really excited to welcome with us today Dr. Steve Paterno, who is Deputy Director at the Duke Cancer Institute, which is at Duke University, where he is Professor of Medicine and Professor of Pharmacology and Cancer Biology, and then also Professor of Family Medicine and Community Health. And he also serves as Deputy Director for the Duke Cancer Institute. Dr. Paterno has a number of honors and distinctions. And as I was reviewing your bio to introduce you, I was like, I don't know where to start. So I might just say that he has several leadership roles across the country, nationally, as it relates to shaping and forming the field of cancer health disparities. Some of the integral work that he's done in this area is co-chairing the AACR NCI think tank on cancer health disparities in 2007. He's been continuously funded by NCI through several different mechanisms, including an NCI Apache and a P20 SPORE and cancer health disparities. So we're really excited to welcome Dr. Paterno to share with us his work in the pursuit of cancer health equity. And I'll turn it over to Dr. Paterno. Thank you.
Thank you, Dr. Hughes Halbert. Much appreciated. We can keep it informal today. I did a quick look at the folks on, and I know so many of you. So if you want to interrupt anytime, just say, "Hey, Steve," and I'll be happy to stop. So today, what I thought I would do is give you a helicopter ride over several different research programs that we have been working on for a number of years now, and all in the context of eliminating cancer disparities in pursuit of cancer health equity. So today I'm going to be touching on a institution level intervention that I had been involved with for many years called patient navigation. We're going to talk about a screening intervention and its impact on policy. And then we'll end up talking about some of the work coming out of our wet lab on the ancestry related biology of cancer, and all along try to emphasize what is increasingly called intersectionality or convergence science, because so many of the determinants of health are interrelated with each other. Just give me a second here. These are my funding sources, R01's P20 Apache and P20 SPORE grants from NCI, multiple grants from the Department of Defense, Prostate Cancer Foundation Challenge Award, funding from the V Foundation and Movender U.S. patents issued and pending, not yet licensed, and consulting and stock relationships with other companies unrelated to this work. Learning objectives to understand multi-level contributors to cancer disparities, describe potential health system interventions designed to achieve cancer health equity, explain how ancestry-related biology might contribute to disparities, and recognizing the importance of precision oncology approaches to achieving cancer health equity. So I thought I'd start with a case presentation. This is right out of of recent case reports from the Duke Cancer Institute. So MB is a 55-year-old African-American male with multiple comorbidities, COPD, tobacco abuse, had a child at age 16, dropped out of high school, works a minimum wage job at a fast food restaurant, has no health insurance, does not own a car, takes public transportation. His mother died from etiologic cirrhosis, was not offered a transplant, causing him to distrust the healthcare system. He was seen in a clinic for the underserved with inconsistent healthcare providers. He was seen in that clinic for an acute visit for dyspnea, wanting a refill of an albuterol inhaler. He had not been seen for 16 months. One of the providers noticed he had lost 25 pounds since his last visit a year and a half prior, asked if he had any cancer screenings in previous visits, including colonoscopy and PSA for prostate cancer. The patient noted, nobody in my family has cancer, so I don't need that, and I don't want to be experimented on like my mother was. He declined lab work and colonoscopy. 3 months later, he presented to the Duke Emergency Department with back pain and leg weakness and was found to have spinal cord compression, a PSA of 1,300, and on radiology, numerous metastatic lesions from prostate cancer. So this case describes this concept, to a certain extent, makes real this concept of intersectionality, where multiple issues are all working together in a negative conflagration to interfere with his desire for healthcare, access to healthcare, and all of these things conspiring to drive an aggressive phenotype of cancer on presentation. So as I move through today, I'll try to cycle back and mention how the work that we do might have impacted this patient's trajectory. So let's talk about disparities. Most of you on this call know all of this. There are government definitions of disparities. The top is the NIH's definition, a health difference that's closely linked with social or economic disadvantage. The NCI has its own definition, adverse differences in incidence, prevalence, death, survivorship, burden, or related health conditions. I have my definition, and that is the unequal burden of cancer among racially and ethnically diverse minorities and the medically vulnerable and underserved. And as all of you know, this problem is both local, regional, national, and global. Well, what do cancer disparities look like among racial groups? These are typical curves that you would find from SEER or ACS. And there's good news and bad news here. You can see that both incidence and mortality rates to cancer overall have been decreasing over time. But you can also see that there are stark differences between different population groups. So the Black populations below that. So you can see that there is a decline in all populations, but the disparity gap has not narrowed. There's some evidence it's beginning to narrow, but at this point, by 2014, it had not begun to narrow. Many times these disparities manifest themselves in the context of cancer aggressiveness, with an earlier mean age at diagnosis, shorter mean survival, more common advanced stage diagnosis, greater risk of early recurrence, and more common metastases. And we also know now that each tumor type and subtype exhibits its own nuances with respect to disparities in incidence, aggressiveness, outcomes, and mortality. One of the best examples of that is in breast cancer, where postmenopausal breast cancer used to be called, back when I was in graduate school, a white woman's disease because of a 17 to 20% increased incidence rate among white women. But that did not draw attention to the fact that there was a 15 to 18% higher mortality rate among women of African ancestry. And we also know that this differs from the picture in premenopausal breast cancer, where African American women present at an earlier age with more aggressive disease. Well, turning to prostate cancer, here are some hard ratios for you to give you a feel for the extent of the disparity in prostate cancer. On the left is the number of new cases, and you can see there's a 1.6-fold difference between incidence and rates between Blacks and whites, and in fact, a 2.8-fold difference in incidence rates between Blacks and Asian Pacific Islanders. But if you look on the right at the death rate, you can see a 5.1-fold difference in death rates comparing Black men to Asian Pacific Islanders. You have to look long and hard through a lot of textbooks to find a disease that shows that magnitude of a population-specific difference in mortality. If you bring the data a little closer to home, this is a risk for prostate cancer in North Carolina and death rates. If you look on the left, those are the panels for whites, and you can just look at the color of those two maps of North Carolina, and then look to on the right side, these are the incidence rates and death rates for Blacks, and you can see a fairly dramatic difference. So this problem is not just national, it is right here in our own backyard in North Carolina. And I think that's the same is true for other Region 2 GMaP South states. Well, the Duke catchment area looks a little bit like this. Duke is a destination cancer program. We see patients from every state in the nation and many, many countries from all around the world. But most of our patients, about 80%, are drawn from the midsection, the mid-Atlantic region, and the red line defines a 80th percentile of our total patients and our patients on clinical trials. And what you can see in the upper right, 24% of that catchment area is African American. Durham is what used to be called a majority minority city, 39.5% African American, and many of these counties are extremely rural and poor. 18 counties in our catchment area exhibit the highest prostate cancer incidence mortality rates in North Carolina, twofold Black versus white, and 2.8-fold in Black versus white mortality rates. Well, what are the drivers? What are some recognized drivers of cancer health disparities? And this comes from a review article that just came out in the Annual Review of Medicine that my team put out, led by Jenny Friedman. And it's just to remind you that we have this intersectionality between societal level cultural frameworks like structural racism, poverty, and implicit bias. We have institutional level environment issues that affect healthcare access. We have neighborhood level social and determinants of health and physical environments. At the center of this, and by putting it in the center, and we're not arguing it's the principal driver, we're just showing that it interconnects with all of the other drivers, is this thing we call ancestry-related genetics and biology, and I will get back to that. But again, these factors and the intersections between them illustrates the need for new methodologies and intersectionality, and increasingly what's called convergent science. Well, in our group at the DCI, we have a vision for community impacting translational research, education and workforce development, impacting disparities. This is from a talk that I give on the road called Trenches, Benches, Bedsides, and Wrenches. And we believe that cancer health, a patient ends up in your emergency room. The case that I showed, health begins in the community. So we want to start our work in the community. We want to translate it into the clinic, and from the clinic to the laboratory, and from the laboratory to the population, and then back to the community via policy, because in order to change the world, we need to impact policy. I mentioned workforce development, and this is just quickly to show that we're engaged in training the next generation of URM cancer researchers. This is our P20 Patchy grant with NCCU. I'm the PI along with Kevin Williams at NCCU. 2 research projects on prostate and inflammatory breast cancer, and a cancer research education program where we have 14 CREP scholars, either grad students or postdocs or early stage investigators being trained in translational cancer research. And we were also privileged to receive one of the coveted P20 Disparities 4 grants. On that grant, we have 2 projects, one on lung cancer, one on stomach, a developmental research program, and a career enhancement program, all focused on raising the next generation of disparities researchers and URM cancer researchers as best as we can. Well, this is also from that annual review of medicine. And here I'm just illustrating to you that today's talk is going to focus on these 2 parts of that continuum: institutional level environment and ancestry related genetics and biology. And when I get to the biology, you'll see that I'll focus my comments on the work we've done around ancestry related RNA splicing. So let's start with the institutional level intervention. This harkens back to patient navigation and my role in the NCI funded National Patient Navigation Research Program, a multi-institutional, 8-institution, large-scale research program that was designed to test the efficacy of patient navigation as an intervention to address cancer health disparities. So why do we need patient navigation? And the answer is actually fairly simple. It's because this is what the health system looks like for all individuals, including highly educated, highly trained individuals, but it's particularly difficult for vulnerable populations. It is a system fraught with healthcare barriers, communications, location of facility, problems with scheduling, underinsured and uninsured patients, perceptions and beliefs, employment loss, wages, language, interpreters, fear, transportation, which is still the #1 barrier to quality cancer care in the country, disabilities, childcare, and literacy issues. So this labyrinth maze really illustrates the need for navigation services. And this comes from a review article that I wrote some years ago. And it lays out what some of these barriers look like. Logistical and structural barriers. I already mentioned transportation and location, distance away from a healthcare facility, system scheduling problems, disabilities, language and insurance, sociocultural issues like fear, perceptions, literacy, social and practical support, communications and attitudes towards providers, and financially related barriers like childcare, adult care, housing, financial problems, employment issues, barriers that are related to socioeconomics. All of these things play out in the real world. And for our case report, Mr. MB, they very specifically impacted the stage of disease at diagnosis and his access to quality cancer care. So Patient navigation was an intervention originally devised by my dear friend Harold Friedman in Harlem, New York. I had the privilege of being one of the earliest adopters of patient navigation and the first to bring navigation into Washington, D.C. when I was the director of the George Washington University Cancer Center for 10 years. Patient navigation is a patient-centered, culturally tailored intervention to help individuals find their way to and through, you'll hear me use this term, it's my term, to and through a healthcare episode and or the complex healthcare system labyrinth and overcoming barriers to accessing quality healthcare across the continuum. And as I mentioned, the PNRP program went on for more than 7 years. There's been almost 70 peer-reviewed publications out of that program. And this is just capturing a tiny snippet of some of the results. And if you just scan down this slide, highlighted in blue, you can see papers showing it was effective in eliminating economic disparities. It improved the receipt of recommended cancer care. It improved timeliness of cancer care. It improved costs and outcomes and significantly reduced delays. And in fact, the list goes on and on and on, showing the efficacy of patient navigation interventions. Well, in the early days of patient navigation, particularly as the model evolved in Harlem, it was focused in its earliest stages on picking up patients at diagnosis and navigating them into treatment as quickly as possible. But in adopting navigation and bringing it into Washington, D.C., we quickly realized that the same barriers that negatively impacted the transition from diagnosis into treatment also impacted patients' ability to access screening, to get screening resolution and an actual diagnosis. Yes, the time span between diagnosis and treatment, but it also negatively impacted the transition either into end-of-life care or survivorship care. So I developed the first, what was called longitudinal patient navigation program in Washington, D.C. And that is the model that I brought to North Carolina and brought to Duke. We have been implementing it both at Duke, but also in North Carolina through my role as the president of the North Carolina Oncology Navigators Association. And this is a paper that just came out last year describing the multidisciplinary longitudinal patient navigation matrix model that we've developed at the Duke Cancer Institute that starts with community-facing navigation on the left. That is integrated through our Office of Health Equity. We have 5 full-time community-facing navigators who are out in the community doing education events, but also navigating patients that need to be screened or want to be screened. In our to and through model, our community-facing navigators get the patients to the front door of the health system and then facilitate a warm handoff to our internal matrix of navigator-trained new patient coordinators, non-clinical navigators who address practical barriers like insurance and transportation and things like that, and also clinical navigators who serve as care coordinators. Every single one of our patients undergoes distress screening, not just at the first admission, but every time they come to the Duke Cancer Institute, where we have a longitudinal assessment of their needs, and our navigators are able to refer each patient to the services that are necessary to meet that patient's needs. So we have been developing this program extensively, and these are some of the folks involved. This is community outreach and engagement and community-facing navigation, our health equity team. Tomi Ekinemieju, who is our associate director for community outreach and engagement. Dr. Angelo Moore is the director of our Office of Health Equity. Nadine Barrett is the director of equity and community stakeholder strategy. And then on the bottom left, you see our team of community-facing navigators, and on the right, our lay patient navigators. Well, as just a quick indicator of impact, You can see here over 2014 to 2018, our navigators, community-facing navigators, reached over 11,000 people in the community. They navigated over 1,700 into screening, and then 242 of those who needed follow-up from a suspicious finding into diagnosis or cancer care. We have an annual men's health event that we put on in conjunction with the Lincoln Community Health Center every year. It was virtual this past year, but in 2019, we served nearly 300 men with prostate cancer screening, fit kits for colorectal cancer screening, on-site skin cancer screening, blood pressure, diabetes, HPV and HIV screening, radon education and testing kits. And every single one of those patients also was connected with a navigator who was able to get them to and through the healthcare system as needed. Working with our FQHC partner, Lincoln Community Health Center, our navigators were, over the course of 2014 to 2018, able to increase colorectal screening rates at this FQHC from 24% in 2014 to over 46% in 2018. And then finally, Dr. Mira Eplein is one of the world's leaders in understanding H. pylori and its role in gastric cancer. She's also heavily engaged in community outreach through a program called DISH, the Durham Initiative for Stomach Health, and working in collaboration with the faith community. And on the right, you see a picture of Dr. Eplein in the middle, speaking with Bishop Godfrey at the River Church. As an example of a typical event, on a Wednesday night, our team went into the River Church. 92 people actually got screened on site for H. pylori. 25% were diagnosed with H. pylori infection. Each person was connected to a navigator, one of our navigators, who connected them to a PCP. They underwent therapy with an 80% cure rate at 6-month follow-up. And in fact, the 2 patients that did not achieve cure, we were able to determine were non-compliant with their medication. So I just showed these as examples of how our navigation program has had community impact. So turning my attention now to screening. This is a study in a transdisciplinary collaboration, intervention, implementation project to address disparities. And here I've only got 2 slides. So if you'll permit me a little chuckling and a little sense of humor here, so bide with me. This is the history of prostate cancer screening over 30 years in 1 slide. And so please smile and bear with me. I'm simply trying to make a point. I'm not engaging in all the nuances. But in 1994, the PSA test was approved by the FDA for screening. And we all know that this saved lives, but it led to large increases in unnecessary biopsies and treatments with major side effects. In 2012, in an now infamous ruling, the USPTF, the US Preventative Services Task Force, formally recommended against PSA screening for prostate cancer. That was based on their analysis of 2 studies, both of which were done in Scandinavia. 3 studies, 2 of which were done in Scandinavia, where there were virtually no persons of color. And one was the PLCO trial. And the PCLO trial, as you know, had a 50% contamination rate and only 3% enrollment of persons of color. Well, what we also know is that evaluation was done too early in the trajectory of prostate cancer, because prostate cancer is generally a slow-growing disease. And when those studies were reevaluated at 9, 10, and 11 plus years after screening, there was a marked and statistically significant bifurcation showing marked survival benefit of prostate cancer screening. In 2019, the Preventative Services Task Force revised its stance, But unfortunately, by that time, there were ingrained practice patterns that did and continue to keep screening rates low. And the downside of that is from 2018 to now 2021, up until just 3 weeks ago, multiple studies keep emerging showing a marked increase in both the number and the percent of men being diagnosed with late stage disease directly attributed to this downturn in screening. Now, in parallel with this, in 1994, I was running a wet lab research program on prostate cancer, trying to understand its biology. In 2002, I became the director of the Cancer Center at GW, and we started launching these interventional programs, including screening programs for prostate cancer in the underserved communities. And in 2012, I was summoned to Howard University, and I walked into a room full of 300 very, very angry people because the announcement of the Preventative Services Task Force recommending against screening had just been made. The reason I was summoned there is I was then the president of the DC Cancer Consortium, which represented all the cancer advocacy organizations in the city. Well, later that year, I moved to Duke and became a governor-appointed advisor to the North Carolina Advisory Committee for Cancer Coordination and Control. In 2014, at Duke, I launched a prostate cancer strategy group to create an integrated longitudinal disease model. And the first thing that we did was to create an algorithm that addressed high-risk prostate cancer, because particularly so many of our patients are of African ancestry and are at high risk for prostate cancer. We launched that screening algorithm in 2016 to 17, and we rolled it out through our EMR, which is Epic, and then implemented it across Duke Primary Care using our Center for Oncoprimary Care. And in 2018 to 19, that algorithm was refined and disseminated and tested through Duke Primary Care. And then just this past year, I was elected chair of that NCA4C. And I'll explain, the next slide will explain why that little point is just a little bit important. So this is from a paper that came out in the Journal of General Internal Medicine. Those of you involved in screening and who have worked with the US Preventative Services Task Force, you might laugh because you'll know that this is a very Preventative Services Task Force friendly journal. So it was actually, we were delighted that they accepted and published this paper. This paper is called Implementation and Impact of a Risk Stratified Prostate Cancer Screening Algorithm as a Clinical Decision Support Tool in a Primary Care Network. And I just want you to see, this was not a small study. This is 50,000 men across 26 clinics, more than 20% of whom were African American. And we were able to show that using this risk stratified algorithm, the percent of patients meeting guideline concordant screening guidelines increased from 49% to 68%, and even higher among African American men. And this led to better precision screening choices stratified by age and risk, with an overall decrease in the utilization of PSA and a decrease in the number of biopsies and subsequent intervention. So we're able to create a precision support tool that increased screening without triggering automatic overtreatment. And this algorithm was then introduced through the North Carolina A4C to the North Carolina legislature last year. It was voted on and adopted as the state of North Carolina's guideline for prostate cancer screening. So just an example of how something that started almost 15 years ago gradually impacted policy. Yes. All right. So I want to be sensitive to time. I'm moving now to the third section of my talk today, called The Biology of Cancer Disparities. alternative RNA splicing as a source of ancestry-related molecular targets in precision oncology and cancer disparities. Now, whenever we talk about race and biology and genetics, I always very quickly want to put this slide up there because this conversation makes a lot of people nervous, and I'm very sensitive to that. I've been doing this work for over 20 years. In fact, my lab in DC was one of the very first labs funded by the NCI to study the intersection between ancestry and the biology of cancer. So I want to quickly relate to you that I completely understand and agree that race in and of itself is not a biological construct. Race and ethnicity are sociocultural constructs. But if you'll permit me just a little leeway here, if you look at two different worldviews, religious and secular, one thing is common to both of those very different worldviews, and that is a very simple statement. We are all out of Africa. And the only difference in any of us, between any of us, in how we look on the outside or function on the inside is simply a function of when and in which direction our ancestors left Africa. So, although race is not a biological construct, racial ancestry as a function of the human diaspora, whether optional, shown on the left, or forced in the context of slavery, shown on the right, that diaspora drove cultural, phenotypic, genetic diversity, and therefore impacted each of our susceptibility to disease, biology of disease, and outcomes from disease. So again, we're not equating race with genetics or race with biology. We're simply saying that race is an indicator of ancestry. Ancestry is simply a function of geography, time, when, and in which direction, of a directionality vector for the human diaspora. So in our lab, we try to understand whether there is an ancestry-related contribution to the biology of cancer using this vision for our lab. And on the lower left, you see we're continually collecting tissue and specimens from patients from different racial and ethnic groups. We have a particular focus in RNA splicing. We're developing lots of molecular constructs and cellular constructs that can be disseminated and utilized by other laboratories, cell lines, xenografts, PDXs. And then Duke has focused much of our activity and much investment on clinical trials that are stratified by race and ethnicity and correlative science. And in the bottom, you see that we have an overarching biobanking protocol called GenCade, which stands for the Genomics of Cancer Disparities. This is a picture of our lab. In the center is Jenny Friedman. She is a cancer biologist and genomicist extraordinaire. She leads the day-to-day operations and is independently funded herself as well by the NCI and the DoD. On the opposite corners, myself and Dan George. Dan is the director of our prostate cancer center and a GU medical oncologist. And then you can see the folks in our lab: April DeVoe, Muthana Al-Abo, Tyler Allen, Sean Pewarski, who's a first-generation Mexican-American, Joab Ordura, and Karen Wegeman. So we try to practice what we preach, and you can see we have a very fun and very diverse laboratory. Okay, let's get to splicing. This is just to summarize my road to splicing. So we started our work around 2005 that first culminated in a paper in Clinical Cancer Research looking at the epigenomics of prostate cancer disparities. We identified differentially methylated genes in prostate tissues from African American and Caucasian men. We then studied androgen receptor genes, target genes in African American prostate cancer disparities. Then we followed up with looking at aggregate gene expression and microRNA, mRNA pairings in African-American prostate cancer disparities. And then the first paper on splicing came out in Nature Communications in 2017. And so I'm going to pick up from there, from 2017, and drill down just on splicing. So this is right out of the NatCom paper. And on the left is Bidar Wang, who then was a postdoc with me and now is a faculty member at the University of Maryland. And on the right is Norm Lee, who was my senior collaborator at GW. And what this just shows is a principal component analysis, not looking at individual genes per se, but looking at exon array. So we did aggregate gene expression and we found some differences, but they weren't really bowling us over. But we right at that time when we were doing these studies, we got a hold of exon arrays from Affymetrix. We put our RNA on there and the results were astounding. And so what you can see in the principal component analysis, in the red circles are African-American samples from African-American patients, in the blue from European American patients, at that time self-identified. And you can see a dramatic bifurcation at the exon level between the two population groups. So this is what gave us the first clue that although we might not be seeing huge amounts of gene changes with very big percent changes, we were seeing dramatic effects at the exon level. So let me do a very quick tutorial because so many of my colleagues, even in cancer biology, don't remember much about splicing. So this just reminds you that at the DNA level, our genes are discontinuous between exons and introns. The exon is the coding region, the introns have to be cleaved out. When the gene is transcribed, the initial pre-mRNA includes both the exons and the introns. But that mRNA has to undergo something called alternative RNA splicing to be able to produce a functional mRNA that can be made into a protein. This was discovered in 1977. It resulted in a Nobel Prize. And what we know now is that alternative RNA splicing drives evolutional biological diversity and tumor-related biological diversity, and that the functions of proteins produced from these splice variants, from a given splice variant, produces a proteoform that can be as different as a protein encoded by a completely different gene. RNA splicing is the way that mammalian cells can make almost 300,000 proteins out of 20,000 genes. We now know that splicing affects every hallmark of cancer: cell cycle, proliferation, metabolism, stress, signaling, immune destruction, angiogenesis, invasion, motility, EMT, DNA damage response, and cell death. And these are the two things that influence splicing the most. On the left, you have what are called cis-acting splicing elements. These are the DNA sequences that reside close to the exon-intron junctions that regulate splicing by virtue of being the binding sites for what are called transacting splicing factors, that particularly a large conglomerate splicing factor called the spliceosome that migrates into the nucleus, assembles splicing. So we start like, what drives race-related alternative splicing? Does it affect biology? Does it help predict risk or survival? And is it clinically meaningful? So on the left is from the Nature Communications paper. And here what I'm showing you is that the DSG stands for differentially spliced genes. And what you can see, over 2,520 differentially spliced genes as a function of race versus 1,000 differentially expressed genes, with about a third of the differentially spliced genes present in adjacent normal tissue, suggested they're carried forward into the tumor tissue. On the right, in a paper that just came out last week, a couple weeks ago, in the journal Lung Cancer, we conducted an analysis on lung cancer, squamous cell carcinoma, and you can see here 4,829 genes demonstrated race-related differential splicing. And of that group, 18% of those genes included drivers, cancer drivers, oncogenes, or tumor suppressor genes. Well, we analyzed TCGA, and these are just the tumor types that we looked at in TCGA. So you can see we're covering the gamut of a large swath of different tumor types. And this was published in a paper that just came out last week in the journal Genomics. We also created, Dr. Al-Aba, one of our postdoctoral fellows, created CanSplice, which is a unique computational tool that can visualize TCGA data and in fact can receive direct sequencing data as well. You can select your tumor type, you can select the type of analysis that you want, splicing aggregate gene expression and how it impacts survival. You can put in the gene name, you can look at every splice variant that you would like, and down in the right corner, you can look at associations with survival as well. So we can make this available to anyone who would like to use it. So this is just showing one way that we analyze that data. Again, this was published in the genomics paper. And this just shows different tumor types, and the red and blue circles each represent a gene. And the red dots indicate differentially spliced genes as a function of race with an absolute delta PSI, that's an arbitrary value of splicing, of greater than 15% difference between blacks and whites, with an adjusted p-value of 0.05. So statistically significant splicing differences. And you can see there's a large number of genes. So how do we start picking out what genes we want to focus on? Well, one way we do it, and one way we've done it in the lung cancer paper, is to analyze these targets by biological processes and by pathway analysis. So we've done multiple levels of pathway analysis, and by looking at biological pathways and signaling pathways, we are able to come up with a number of top-tier genes that undergo race-related differential splicing that we're interested in following up on the biology. A second way that we do this computationally is we can take these lists of race-related differentially spliced genes among different cancers and overlap them. And here we're simply asking or speculating that if a couple of genes show up as differentially spliced in a race-related or ancestry-related fashion, common between different major tumor types like breast, colon, and lung cancer and prostate cancer, maybe those genes are important. We're just showing you that in fact there are 18 such genes at the bullseye of this Venn diagram. And I've just put in blue some interesting candidates that we are following up. A RAS suppressor type protein, gene involved in ubiquitization and protein degradation, and a number of others as well. And another way that we analyze these large number of genes is to link them to survival probability. In this example, we're looking at prostate cancer, so we're looking at disease-free survival probability. And what you can see is that in the blue circles, you have differentially spliced genes associated with poor survival probability in whites, where it's better in African Americans. And in the yellow, you see the opposite, where poor survival is in African Americans, but better in whites. So by doing these kinds of survival analysis, we're able to identify genes that we think are playing a role in driving the biological difference that we discussed earlier, particularly in the context of tumor aggressiveness. So we've identified a large number of race-related alternative RNA splicing differences between Black and white cancer patients. A large number of these genes overlap across cancer types, and that they were determined to function in oncogenic pathways and associated with survival. Well, getting to the biology, I'm being cognizant of the time here. This comes from the Nature Communications paper, and it's just one example of two that I'll give you. we were able to discover that the PI3K delta isoform, the delta isoform of PI3K was differentially spliced between black and white prostate cancer patients, and this is based on an analysis of biopsy tissue. And in fact, most African American prostate cancers had a short form where exon 20 was spliced out, whereas most of the samples from white patients had a long form that include exon 20. So we did a number of studies where we either cloned the short form or the long form and overexpressed them, or we created siRNAs to knock down one form or the other. And in the lower left, what you can see here is in a prostate tumor cell line derived from white patients, when we used an siRNA that knocked down the long form, including exon 20, invasiveness of those tumor cells went down. But when we did the same thing in a cell line derived from African American patients, invasiveness went up. And this gave us a clue that exon 20 was playing a role in the more aggressive biology or contribution biology of this of PI3K delta. When we knocked down the short variant, we were able to decrease the invasiveness of the prostate cancer cell line derived from African American patients. For the sake of time, I'll skip over panel C. That's just an in vivo study showing what happens when we overexpress the short form versus the long form. But what you can see in the lower right corner is that we went back into TCGA and we analyzed it for the ratio of the short isoform over the long form. And in every case, whenever that ratio was higher, that was the short form was more prevalent, survival was worse. So that began to show us that this gene was actually playing a clinically relevant role, and this splice variant was playing a clinically relevant role in the biology of prostate cancer. We also, from that study that we did where we overlapped differential splicing among different tumor types, these are some of the top genes that showed up. And you can see at the top, insulin receptor, CD44. Many of these are very famous genes involved in invasion and metastasis. One of our postdoctoral fellows became interested in LMO7, which is a limb domain protein that modulates actin signaling. Utilized CRISPR to very specifically cut out one exon, the exon that was playing, that was identified computationally as the exon playing a role in the differential expression. We clipped out that exon, and then in a prostate tumor cell line, and what you can see on the right is when we clip that exon out, both migration and invasiveness of DU145 prostate tumor cell line increased. So once again, not shutting down or changing the expression of an entire gene, just clipping out a very specific exon. We went back into TCGA, and sure enough, if you look at either low expression of LMO7 or simply skipping of exon 12 skipping, in both situations, survival is worse in patients that are missing that exon. So once again, a clinical correlate to a biological observation. I mentioned to you early on that we that one regulatory aspect of splicing is cis-acting DNA sequences, and I'm just showing you here that we've done an analysis for SNPs for variants. Genetic variants in the splice regulatory regions at the DNA level. These two papers focused on stemness-associated genes, and in fact, we were able to demonstrate that mutations, SNPs and RNA regulatory variants of these genes correlated with racial disparities and susceptibility to prostate cancer, that using the multi-ethnic cohort and in using PLCO, we were able to correlate splice regulatory SNPs with survival. And we're about to submit a new paper looking at splicing-related SNPs in lung cancer. And this was in a large-scale GWAS-like study of over 30,000 cases and 50,000 controls. And in fact, we identified 3 SNPs in key genes that impact the biology of lung cancer in this case. We also have now race-related splicing SNPs in known prostate cancer-related genes, and then we've done a genome-wide scan as well. And you can see there are some key cancer-related genes in this group: ATM, mTOR, and KLK3, by the way, is the gene that codes for prostate-specific antigen. And even in that gene, we found a race-related splicing SNP. For the sake of time, I'm going to not spend much time on this. I just want you to know that there's another half of our lab working on the therapeutic side of splicing. And this is just showing you that there is an intervention that has been developed called splice switching oligonucleotides that we have been working on. And this is an example of a splice switching oligonucleotide that targets ARV7, which is a splice variant in prostate cancer that codes for the evolution of resistance to hormone therapy. And you can see that our splice switching oligonucleotide actually restored sensitivity to enzalutamide treatment by decreasing the expression of the pathological splice variant while maintaining expression of the physiological splice variant. We also have race-related DSGs in multiple immune-related pathways, including adaptive immunity, NF-kappa B signaling, and innate immunity, and in the inflammasome pathway in lung cancer, and we're beginning to study each of these. So these are race-related differentially spliced genes impacting the immune system. Okay, so I'm going to end here, and I just want to quickly show you the results of exciting results from 2 different studies of clinical trials in prostate cancer where they were purposefully stratified by race. This is the PROCEED trial that was coordinated in Louisiana by Oliver Sartor, but a trial where Duke was actually the highest accruer, patient accruer. This is a CYP-T cellular immunotherapy trial. What you can see in this paper, that the overall survival rate of PSA-matched Black and white patients treated with Sipuleucel-T, African Americans, overall survival was improved by 9.5 months. So in that trial, there was a better response to cellular immunotherapy. And this is a paper that's in press right now. And this is a prospective trial of abiraterone plus prednisone, stratified by race, conducted here at Duke. Dan George is the PI. And in both of these trials, Black men came into the trial with worse disease almost uniformly, but responded better to therapy. So we're very excited about these results, and we can't imagine results that could be any more exciting in the context of working hard to achieve cancer health equity, and that trials that really underscore how important it is to take race, ethnicity, and potential population level differences into account when we're designing clinical trials, and being very specific, purposeful, and intentional to over-select for persons of color who are usually underrepresented in clinical trials. So this summarizes what I've told you about splicing here today. This comes out of a commentary we published in Clinical Cancer Research, just reminding you that tumor cells that come from different populations, ancestral populations, can exhibit SNPs in the genes that regulate splicing, differences in the transacting splicing factors, differences in how the pre-mRNAs are actually processed and spliced, and differences in the proteoforms that are produced. they can serve as biomarkers, they can produce neoantigens, and they can serve as targets for RNA and protein targeted therapeutics. So it takes a lot of people to do these kinds of studies. I've tried to point out the folks in the lab and all along, but these are all the people that have contributed to this work over the last 9 or so years. And it takes a lot of money to do these kind of studies. And so these are our funders, the NCI, Prostate Cancer Foundation, Movember, DOD, Murdoch, Peter Michael, and the V Foundation. So sorry I went over time. Thank you for your attention.
Thanks so much, Steve. We are at our time, but I do want, there was I think a really important question in the chat that I just wanted to give you an opportunity to speak on. The question is, who pays for the patient navigation service? That's such a comprehensive program. And I think there's interest among all of us of how, what resources are available and how that's paid for.
So that's a great question. If any of you are available, you'll hear more about this on Friday. The North Carolina Oncology Navigators Association is holding its annual meeting, and Tracy Battaglia, who chairs the National Navigation Roundtable, will be addressing some of this. You know, for decades, the majority of navigation programs in the United States were funded off of grants. And that was actually a very difficult, complicating factor, because the grant would run out and the program would go away. I was keenly familiar with that when I moved to North Carolina. So I actually made it part of my recruitment package, if you will, by the Duke Cancer Institute, that we were going to, that Duke was going to make a hardwired commitment, longitudinal commitment to the Office of Health, to form the Office of Health Equity. And so at Duke, our navigators are all hardwired. Their positions are all hardwired. We do seek out grants, and if we get grants, we can offset, you know, some of those costs. But if the grant goes away, the position remains and the persons remain.
Thank you.
And our health system has bought into this because we have shown that it actually saves money on the downstream.
Got it. Well, thank you so much for a fantastic presentation. Just because we are a little bit past the hour, and we want to be respectful of your time and everyone else's time, I just want to thank you for sharing your information about your program. It was fantastic to hear about all of the exciting new discoveries that you and your lab have made, and so we're really appreciative of you presenting today. And
I am reading the comments now, and
yeah.
For those of you, a couple of you have asked questions. You and I, we have been in email contact, so feel free to email me, and I'll answer them offline as well.
That was my last question, so to make sure that people could get in touch with you with follow-up questions.
I can see questions from Dr. Wheeler and Henderson, and they know how to find me. So.
Okay, great. Well, thanks everyone for joining our seminar, and thank you again, Steve, for a fantastic presentation. Okay, we'll sign off now. Thanks.
Thank you all.
Bye.